Today we are going over mitosis notes. Okay? There are two types of cells that we need to know about from the start. The first type we're going to talk about are somatic cells. Somatic cells are your body cells. Skin cells, brain cells, cheek cells, intestine cells. These cells reproduce by mitosis. In mitosis, the chromosome number after the cell divides is the same as the chromosome number before the cell divides. Okay? Sometimes you'll see this in questions. For example, humans start with 46 chromosomes in their cell. After mitosis, the new cells also have 46 chromosomes. Sometimes described as 2n to start, so you'd have 2n at the end. And then these 46 chromosomes are also sometimes described as diploid. So if you started with a diploid amount, you'd end up with a diploid amount. The next type of cells we talk about are gametes. A gamete is a sex cell, okay, sperm cell or egg cell. These cells reproduce by meiosis. Okay, mitosis and meiosis look very, very similar, so they sometimes get mixed up. Mitosis happens with body cells, meiosis happens with sex cells. In this case, the chromosome number after division is cut in half. So for humans, their starting cell has 46 chromosomes. But after meiosis, making a sperm cell or an egg cell, those cells only have 23 chromosomes in them. Okay? So sperm cells have 23 chromosomes, egg cells have 23 chromosomes. When a sperm cell and an egg cell combined, they then make a full 46 chromosome set. Sometimes you'll see 2n to 1n or 2n just to n. And then if we start with a diploid amount and we half it, that's now called haploid. Okay? A little bit more about these two methods of cell division. The first one is mitosis. This is cell division producing daughter cells with identical chromosome number. So let's start by talking about daughter cells. Daughter cells is the term that's used for cells that are made from mitosis or meiosis. And in this case, the daughter cells or the cells that are made have identical chromosome numbers. So no matter what organism you're talking about, the number of chromosomes at the beginning are the same number as the chromosomes at the end. The second method of cell division is meiosis. This is when you produce daughter cells with half the chromosome number. This only, again, happens in sperm cells or egg cells. Let's flip to the next sheet. And we'll talk about the life cycle of a cell. So the life cycle of a cell has a few parts. The first part is what's called interphase. Interphase is actually the time in between the cell dividing. Number two, the second step is actually the cell division step. Okay. This involves mitosis and something called cytokinesis. So mitosis is actually technically the division of the nucleus. We'll talk about the 4 stages on the next page. Cytokinesis is the division of the cytoplasm. Now, when you have cells dividing, and the cytoplasm eventually divides, cells either form a cell plate or a cleavage furrow. Cell plates happen in plant cells. Cleavage furrows appear in animal cells. And the last phase of the cell cycle is the G0 phase, and this is when the cell exits the life cycle phase. So in this, we actually exit the cell cycle. Okay? There would be no more cell divisions. Okay? This does happen in your nervous system cells, where they never go through another round of the cell cycle. They actually exit the cell cycle and stop dividing at maturity. Let's flip to the next page and we'll go through all of the phases or stages of that nucleus dividing. So in this section, we will be looking at the different stages of mitosis. Okay? The first one we start with is interphase. Interphase is not technically a stage of mitosis because it's just basically the phase in between the cell actually dividing. So sometimes you may not see interphase listed as a stage of mitosis. In interphase, the chromatin, which is that unraveled DNA, replicates. So that's when the cell is making a copy of its DNA. The cell grows a little bit and then rests up for the impending division. This is a picture of a plant cell in interphase. We are actually going to draw a picture ourselves. Our pictures will be animal cells, so we will be drawing circles. So if this is my cell membrane that I just drew, we need a nucleus, and the chromatin just sort of looks like it's lightly colored in. Okay, there's no distinguishable chromosomes. The next stage of mitosis is prophase. The replicated chromosomes that happened in interphase are now actually pairing up, and they become a little bit visible. The nuclear membrane disintegrates, so you start not seeing the nuclear membrane as much, and eventually by the end of prophase, there is no nuclear membrane at all. And something called spindle fibers start to form. So we're going to draw our circle again. Okay. I'm going to draw our nuclear membrane in the process of disappearing. So I like to draw the nuclear membrane with some like holes in it, basically. You'll see in the plant cell to the right these thicker black lines in the nucleus. Those are the visible chromosomes. Okay. For our picture, we are going to do X's. I'm just going to draw 4 random X's in here. And then in terms of the spindle fibers, you just got, they sort of look like hairs growing out of the side. That's our prophase. The next stage is metaphase. Metaphase is when the doubled chromosomes attach to the spindle fibers and line up at the equator or the middle. So what's awesome here is that metaphase starts with an M, and we line our chromosomes up in the middle. So metaphase, middle. Let's draw our animal cell again. You see in the plant cell, these chromosomes are nicely lined up at the equator or the middle of the cell. We're going to do the same thing. So I'm going to take my 4 chromosomes, line them up in the middle. I do mine top to bottom. Sometimes you'll see them left to right, and that's fine. Okay. Now, if you remember, animal cells have centrioles, which aid in cell division. And now that we've moved into metaphase, the spindle fibers will come out of the centrioles and attach to the chromosomes. In this stage, I'd like us to label what's happening in the cell. So right here, this is the centriole. Sometimes they're circles, sometimes they look like cans, basically, like cylinders. Okay. This is the spindle fiber. And this X is the chromosome. Okay, just some things we need to know in terms of labeling these cells. The next stage is anaphase. Anaphase is when what we call identical sister chromatids. These X's, these chromosomes that have paired up, actually separate and move towards the opposite poles. So we still need our circle for our cell membrane. Okay. You see in the plant cell, these chromosomes are being pulled apart. So I like to start my drawing with my centrioles and then have spindle fibers coming off of them. Once you've got your spindle fibers set, each spindle fiber has half of the X chromosome attached to it. Okay, so half of that letter X looking chromosome is attached to the end of each spindle fiber. Now, we talked about metaphase equals middle. In this case, anaphase means chromosomes are being pulled apart. Anaphase, chromosomes get pulled apart. And the next and final step is telophase. This is when the half of the chromosomes reach the opposite poles. Cytokinesis, that division of the cytoplasm, begins, and new nuclear membranes form. So you can see in the plant cell, the chromosomes are at the opposite poles. Okay? There is a faint line here in the middle. Please label that the cell plate. The cell plate only happens in plant cells. For us, we are going to draw an animal cell that's being pinched in the middle. Okay? We're going to draw our nuclear membranes reforming and our chromosomes inside. And lastly, the indent is called the cleavage furrow. The cleavage furrow is only in animal cells. Okay, these are the phases or stages of mitosis: interphase, prophase, metaphase, anaphase, telophase. If you go back up to the top of the sheet, in terms of the stages of mitosis, I find it easier to remember them if I use the first letters of each phase. Okay, for example, ip-mat is a good way to remember all of the stages of mitosis: interphase, prophase, metaphase, anaphase, telophase. Ip-mat is all of the stages in order. It's now time to look at the last page of notes. This is just a general review of the results of mitosis. Okay, again, results of mitosis. So please make sure you realize that this is mitosis. Two daughter cells, you've got identical nuclear information, so the same amount of chromosomes you started with. Each daughter cell does contain about half of the cytoplasm and organelles, but as they grow, all of that will be replenished. Okay? Mitosis is also a form of asexual reproduction. Okay? We will get into some more details about that, but asexual reproduction includes budding, binary fission, regeneration. Okay? And then some differences in plant cells and animal cells based on the mitosis. So, plant cells, we've got the cell plate. Animal cells, we've got a cleavage furrow. Plant cells, there's no centrioles at all. Cleavage furrows, the animal cell does have centrioles. Asters, sometimes if you've got a centriole and you've got the spindle fibers coming off, the asters are just little fibers in the back.